So we can submit that. Okay, it's taken a little while. It's finished. And we're going to look, and this tells us, you know what? No signal peptide. So it's looking along here. It's only looking in the first, what would this be, 40 or 50 amino acids? Because that's where the signal peptides are. They're at the amino terminus. We don't see anything. It would have to go above this little line right here, which would be the threshold for a signal peptide based on what we know about signal peptides across various proteins. And this one just doesn't have any of the amino acid signature that would be required for a signal peptide. So we're pretty confident in saying this protein probably doesn't have a signal peptide. Does that mean it's not targeted to a cellular compartment? No. It's got to be targeted. It's in a membrane somewhere. It's got to be going to a specific membrane. It just doesn't bear a resemblance to a targeting sequence that we normally see in proteins. So there's all kinds of these things. There's another one, signal prediction. That kind of does the same thing that the Signal P 4.0 server did. We can look at glycosylation. Is there a possibility for glycosylation of this protein? Just do the same thing. Just paste it in. You can see this site and the other one are run by the same group. So it's finished, and here we go. Warning, this sequence may not contain a signal peptide. Okay. That's important for glycosylation. Why? Why is it important? Why would they tell us that? So we've got some sequences that might be glycosylated here. Why is it important? I mean, this is sort of cell biology. It's not really genetics. So what kind of protein would be glycosylated? Where does the glycosylation occur? Extracellular domains, right? That's where you get glycosylation. So you'd have to have a protein targeted to a membrane, and specifically the cell membrane, in order to really truly be one that's glycosylated. So we don't see a signal peptide on this, so that probably means it's not in the cell membrane. Although it still could be, again, this is just a hypothesis. But, so anyways, we'll go back. We can look at phosphorylation. Put it in here. Where's it? Oh, there's it. And we can see, is this protein likely phosphorylated? Well, we saw in the ELM that there were phosphorylation sites, right? So there should be phosphorylation sites in here. And that's what we're seeing. It gives us the predictions for 3 different phosphorylation sites. Serine phosphorylation sites, so serines that are located in the protein. So here's a little peptide for the protein. Here's the serine in question, and the question is this serine phosphorylated? Is it likely to be phosphorylated? And so when it has a certain score, then it's predicted as being a phosphorylated serine. Is it really phosphorylated? We don't know, but it's, again, a prediction. What would phosphorylation do to a protein?
Either activates it or inactivates it.
Right, turns it on or off. Phosphorylation is a very important way for proteins to be regulated, to be turned on or off. You phosphorylate a protein, you either turn it on or turn it off. It changes a slight conformation in that protein's structure. So we can see there's a bunch of serine predictions for phosphorylation sites, threonines, and tyrosines. So we can get those, and here's the set of predictions. So obviously, a whole heck of a lot of predictions, right? This protein looks like it's phosphorylated all over the place. Are all of these phosphorylated? Probably not. right? There may be one or two on this protein that's truly phosphorylated. Another one that I think is a really cool one is the fingerprint scan. So this is, the ELM is run by the European Molecular Biology Laboratories. You see up in the left-hand corner. They have they're just really good about having all these online databases that are just superb. So let's run fingerprint scan. So this is going to tell us little domains that have maybe some functionality. So it comes up here and it looks at all of these different fingerprints. So it tells us which kind of fingerprint we're looking at. So this one right here, let's see if I can open it in a new tab. It's going to tell us about this fingerprint, what it does. It gives us information. Human genes containing triplet repeats can markedly expand in length, leading to neuropsychiatric disease, blah, blah, blah. Tells us about this region right here. So here's a region of this protein that could be important in disease if it was in humans. So, and it's shown us the region of the gene, of the protein, where this is important. Let's see if I recognize any of them. Yeah, these are probably all interleukin 1A. So we can see here that there's a interleukin site, interleukin fingerprint on this protein. So there's all kinds of things we can learn through these databases. We can learn a heck of a lot about gene function. I want to try to sell you on bioinformatics. Bioinformatics, all I'm doing is giving you an incredibly brief taste of bioinformatics. There's so much more in bioinformatics than just looking at these few things. It's not all about predicting protein function either. And last year, we hired a new bioinformaticist. His name is, oh my God, I can't remember Ahrensberger's last name. Ahrensberger, thank you. Peter Ahrensberger. Yeah, I was the head of the search committee, so why do I forget his name? But in any case, so he's a bioinformaticist, and he's going to be offering bioinformatics either in the winter or the spring, and I would highly recommend taking his course if you're interested. I've got to tell you, when we were interviewing candidates, the first part of the interview was a Skype interview, which is not all that comfortable for the candidate to get on and interview, and he was just amazing. We all just were blown away by his ability to teach us during the Skype interview. And so I think he's going to be a fantastic instructor here, and so I think he's going to be great in his bioinformatics class. So look for that. Maybe he doesn't want me, leading too many students to him at this stage. But, okay. I just want to take a couple of minutes and talk about some things that people do with genes once they've done bioinformatics. What other sorts of things can you do with genes in a sort of a brief way, just to learn something about them? And one of the things that people do is to make reporters, to study the function of the gene. So what does this mean, a reporter? Does anybody know? Yeah.
Is there like a sequence tag that can be accessory to a protein, doesn't interfere with its function, but
Right.
you can track it, intercept in vivo?
Right. So it's a signal that is put onto the gene and winds up on the protein. And that can give us an idea of where and when the gene is expressed. Where and when that gene is expressed. So, reporters are one way to do that. Antibodies are another. So if you have a gene that you have discovered and you want to learn something about it, you can get a little piece of the protein, and it doesn't have to be a big piece of the protein, it doesn't have to be the whole protein, but you can get a bit of it, and you can send it off to a company that will produce an antibody to it. So what they do is they typically inject it into some organism, a chicken, a rabbit, a mouse, a rat, something like that. That organism will develop an immune response to it, produce antibodies to it, and you will then they collect the blood from the from that organism, and the blood will contain the antibodies. You purify the antibodies, and then you can do immunofluorescence and look at the location of the protein within the organism that you are that has that gene. So antibodies can tell you just in the same way reporters can. These two are very similar. One other thing that people do sometimes is they want to know where a gene is transcribed. So they want to know where transcription occurs. And so what they do is what we call in situ hybridization. In situ hybridization. And you make up little pieces of DNA that hybridize to RNA. Sorry about going so far down on the board. So you make up little pieces of DNA that hybridize to the RNA, and you label those pieces of DNA. You put something on that can tell you where they are. And let me just show you a picture of an in situ